Hello, good morning everyone. We are going to discuss the active transport and the vesicular transport in this lecture today. So last class, that was day before yesterday, we stopped with the concept of osmolarity and osmolality along with the osmolarity gap with its clinical implication. That was a part of the passive transport, right? Now comes the other part of the transport or other type of the transport, which is the active transport. So by definition, if we go by definition, active transport at first is like it is the mode or type of transport that essentially requires energy. And the type of energy here that is needed is nothing but the ATP. That is the definition of active transport. Now, this active transport is again of two types mainly, which is the primary active transport and the secondary active transport. So traditionally, we have always heard and understood and studied about this two type of transport. But then one recent addition was the transportation in which a vesicle is needed. So that way again another type of active transport has been added, which is also considered to be a type of active transport as the vesicular transport. Okay, so this transportation we will study one by one in details. First, to start with primary active transport. Now, one very interesting thing and one easy way to remember the name and the examples of this primary active transport is that this transport, it needs ATP and obviously it needs a carrier protein also, right? So, all the pumps, all the carrier protein that has a ATPase is a active transporter, especially primary active transporter. So, the outstanding examples of this type are sodium, potassium, ATPase. Okay, hydrogen, potassium, ATPase. Calcium, ATPase. So, look here, as I told you, in all the pumps of this primary active transport, there is this energy giving carrier protein, which is the ATPase. Another interesting yet important characteristics of this primary active transport is that here, that means in this primary active transport, here the ATP is directly derived from the pump. Which pump? That is the ATPase pump. So if it is the sodium potassium ATPase pump, this ATPase is an enzyme, PASE. S means enzyme, right? And the function of enzyme is to cleave something, break something, digest something. So here the ATP is directly derived from the pump. Meaning what? This attached pump, it will break the ATP into ATP plus IP, which is the energy provider. Means this IP will ultimately change the configuration and give energy to the pump to carry out the transport. This is like the energy part of it. Okay. Now, going by the concentration part of it, concentration gradient. Like from what concentration to what concentration molecules will be pumped? Here, since we are using a carrier protein as well as we are using the energy in form of ATP, what will happen is that the molecules will be transported against the concentration gradient. Let's emphasize on the term against. Here, what does against mean? Against mean from a region of lower concentration to a region of higher concentration. Against means from a region of low concentration to high concentration, which is just the opposite of diffusion that we studied. To understand the functioning or work or the mechanism of this primary active transport, we will take the example of the sodium potassium ATPase pump. Let us understand and study what is this sodium potassium ATPase pump. First reason why this pump we are going to study, there are so many other pumps also, right? Why this one only? Okay. First, because this is the most abundant pump in the body. Means our body has got millions of cells, and the most abundant, the most common active pump that is present is the sodium potassium ATPase pump. This is the first point. Second is this pump consumes lion's share of energy produced. Okay. Lion's share means maximum. So it consumes maximum ATP that is being produced by out of all the pump, this pump consumes the maximum ATP, maximum energy. Okay. Now, what are the features? What are the features of this sodium potassium pump? The salient feature of the sodium potassium pump. First, we start with like this sodium potassium ATPase pump, which is also an exchanger. This is an electrogenic pump. This is one feature. Second feature is that it means this pump consumes 24% of energy required in neuron, like when the neurons are also cell, right? They're the brain cell, they're the units of the brain cell. So neuron also needs energy for its functioning. So out of all the energy that is used by the neuron, this sodium potassium ATPase takes about 24% of that energy. I repeat, out of total energy that is being consumed or utilized by the neurons, the sodium potassium pump in the neuron, they consume 24% of it. Okay. And about 70% of energy in the periphery, like the peripheral cells, like other than the brain, the neurons wherever it is innervating from the brain, originating from the brain and spinal cord, whichever structure it is, or innervating, they are 24%. And in the peripheral cells, other cells of the body, about 70% of the energy that is being consumed is by this sodium potassium ATP. Okay, that is the energy requirement. Physically, how does it look? We'll draw it also. It is a heterodimer. Now, by the term dimer, we know that it has got two subunits. Heterodimer containing alpha and beta subunit. Okay. Another one is that its alpha subunit is like transmembrane in nature. Like the alpha subunit, it goes all the way across the ECF to the cytosol. We will of course draw and then understand the diagram. The beta subunit is mainly for glycosylation. It has got the sites where only glycosylation happens, but the transportation, the carrying part, it happens by the alpha subunit. The alpha subunit has binding site both facing and touching the ECF and ICF. So now with this diagram, we will understand what actually the configuration looks like. Okay, say this is our cell membrane. Okay, so this is the cell membrane and this is the ECF and this is the cytoplasm, obviously the ICF part of it. So now if I draw this, the beta subunit is something like this one. Of course, it's a hypothetical diagram, but this is easy for us to understand its functioning. Okay, this is the beta one. The alpha one it is postulated to be something like this. Okay, little bit more gap, something like this one. Okay, we write it out. This is the beta subunit and this is the alpha subunit. Now, this beta subunit, as discussed, it has got glycosylation site. It has got glycosylation site, which is reported to be mainly 3 in number. 3 glycosylation, like 1, 2, and 3. It is reported to have 3 glycosylation site. So we write it down. This is like the 3 glycosylation sites. Now this alpha subunit, it has got further portions or further areas which is specific for specific binding of the substances. Okay, so here there is a binding site. We'll discuss what binds into that, and here also there is a binding site. Okay, so this is called the site 2, and here there is something that is called as the site 3. Here there is something that is called the site 1, and then we have 4 and 5. Okay, please remember this is a hypothetical diagram, but it is mainly used for our better understanding. Okay, now here physiologically, I'll come back to this. The main function of this pump is to pump sodium out from ICF to ECF, like rather specifically 3 sodium molecules out, and pump 2 potassium molecules from ECF to ICF. So basically this is doing the function of an exchanger, function of a transporter, right? It is pumping 3 sodium from inside the cell to outside the cell and bringing in 2 potassium. So actually 1 plus or positive ion is getting pumped out extra to outside. Thus the the coupling ratio of sodium to potassium is 3 is to 2. 3 is to 2. So let us understand with the diagram. Like here from inside the cell at like site 1, 3 sodium will bind. This is the binding site. This is the binding site for sodium. Here 3 sodium will bind, which will be pumped out to the exterior, that is the ECF, and from the other side, this is like the binding site for, this is the binding site for the potassium. So 2 potassium will bind to get pumped inside the cell. Now, apart from that, just very close to this potassium binding site, you can see that this site is actually the covalent binding site. Okay, and 4 is the phosphorylation site and 5 is the ATP binding site. So, if we write it down, it would be like the binding site on alpha subunit facing the ECF are for facing the ECF are for two potassium molecules and the cobain. Now, what is cobain? It is a derivative of digitalis like decoxing and all. These are all cardiac glycosides. Okay. And the binding site on same alpha subunit facing the ICF are for three molecules of sodium, that is the binding site 1. Then we have for ATP binding site and then phosphorylation. Okay, so these are the binding sites. Now, what are the hormones that affects the sodium potassium ATPase? Does all hormone have the same effect on the sodium potassium ATPase? Hormones are what? Hormones are nothing but they are the protein having some specific function that initiates or accelerate a function. Okay, so it is seen that aldosterone, thyroid hormones and insulin, all these three hormones stimulate they stimulate and increase the sodium potassium ATPase pump activity, that they will increase the speed, the rate at which the sodium potassium ATPase pump is bringing in potassium and throwing out sodium out of the cell. And this pump is present everywhere in all the cells, in all the structures we have, our brain, our heart, our lungs, our peripheral cells, kidneys, everywhere, liver, everywhere, somatic cells, everywhere. Okay. The cell that inhibits this sodium potassium ATPase is like dopamine. This inhibits the sodium potassium ATPase pump activity. It slows down and it does by the method of phosphorylation. Phosphorylation. Okay. What is the clinical implication? What is the clinical aspect? The clinical aspect stands out to be the presence of the cobain binding site. So, cardiac glycosides like digitalis, okay, they bind on the cobain binding site. And once they bind on the corbing binding site, immediately what they do, this cardiac glycosides, they inhibit the sodium potassium ATPase. So as soon as they inhibit the sodium potassium ATPase, what will happen? There will be increase in the contractility of the cardiac muscles. So it is used to prevent a failing heart. When the heart is pumping slow, heart is weakening, heart is almost going to stop pumping, that's when this kind of inhibitors are used so that the contractility is increased and the heart is stopped from failing. Okay. A recent study is published in few literatures and books, which is about a steroid derivative drug, goes by the name istaroxime. Now, this is a new finding which is reported. This istaroxime, it is seen to inhibit the sodium potassium ATPase. Okay. Now, because of this characteristic, this drug is used to increase the the ventricles of the heart's ventricular contractility. But also the limiting factor is that how this particular drug is actually functioning, that mechanism or the mode of action, this is still not reported. It is yet to be found out. Okay. So we have understood the primary active transport with the detailed study of the sodium potassium ATPase. I hope there is no confusion in it. If you have any, you can ask me. Let me just check. Okay. Now, next we move on to the secondary active transport. Secondary active transport. See here, this is also an active transport, right? It is secondary active transport. So, by definition, if we go, yes, it is an active transport. It obviously requires energy. True, it requires energy. Then how is it different from the primary active transport? In terms of the energy availability, in primary active transport, what did we see? The pump, that ATPase, the pump itself broke down, right? It broke its ATP and provided the ATP and IP. But in case of secondary active transport, this does not happen. Here, the energy ATP is indirectly attained from sodium potassium ATPase. Okay, how do we understand that one? Okay, now see, The outstanding examples of this secondary active transport are like sodium glucose co-transporter. Then we have sodium iodide co-transporter. We have sodium potassium chloride symporter. Like all these things are getting transported together. Then we have also exchangers like sodium calcium exchangers. Okay. We also have anion exchanger, chloride and bicarbonate exchangers, which is also known as anion exchangers. They are present like in, it can be anion exchanger 1 or 2, all this. Okay. Now, for our understanding purpose, let's draw the sodium glucose co-transporter. It is most abundant in the renal tubules, specifically in the proximal convoluted tubules. So, if we are drawing this one, okay, just for our understanding purpose, say this is our, you know, like proximal convoluted tubule, one like cell. Okay, so here there is this symporter or co-transporter, which is the sodium and this glucose importer. Okay, by which sodium and glucose are entering where into the renal tubule, and it is entering. This is the lumen. This is our ECT, and this is our plasma, the blood. Okay, so this is the secondary active transport example. This is a mechanism by which the sodium and glucose are entering together. Now, as I told you here, the ATP is indirectly attained. So, who's giving the ATP to them? Here in this, we have not seen any ATP is attached to the sodium glucose transporter or symporter or co-transporter, whatever we call it. Here, again, few minutes back, what did we study? In all the cell, there are numerous number of this sodium and potassium exchanger, right? So this is that sodium potassium ATPase, sodium potassium ATPase pump that is present in all these cells. They break down and they provide energy for this secondary active transporter, for the symporter. Now, there are other concentration gradient and everything working in it, which we will study in our renal physiology. For now, you just remember that in all the secondary active transport, of course, we need energy, but that energy comes indirectly from the sodium potassium pump. Okay, this is what it means. In some classical way of definition, in olden times, it was also told that like primary active transport is primary active transport. Okay, secondary active transport means it was earlier generalized. It was generalized that when any molecule, whether it is cation, anion, or any neutral molecule, anything is co-transported with sodium, it is called secondary active transport. Now, this definition was true till like 90-95%. But again, you see, we have mentioned about some anion exchangers and all, like chloride and bicarbonate exchanger. This is also an exchanger which is very much present in the RBC as well as in the renal cells, renal tubular cells. So, it is not that only the molecule which get transported either together or in exchange with sodium are the secondary active transporters. There are very few, but still there are some which are exception to this also. Okay, if we, to summarize the thing, if we draw the examples, it will be something like this. So, this is our ECF. This is our cytoplasm. And this is the cell membrane. If we are to draw, like this is a importer. Okay. The transporter, the examples of the secondary active transport, both co-transporter as well as exchanges. Just have a look at this. So here we can say, like our first example, sodium, glucose, co-transporter. Our second example, we have sodium iodide co-transporter which is present in the thyroid follicular cell. Our third example, sodium, potassium and chloride importer. Then we also have exchangers, means going in opposite direction, like sodium entering and calcium going out. Sodium calcium exchangers. Then we also have this anion exchanger. Chloride going in and bicarbonate going out. So here you see, these are the various type of the secondary active transport system. Okay. Talking about the clinical links. Clinical links. What will happen if this secondary active transporter don't act efficiently, or if they stop acting the way they are acting? What will happen? First example, there are numerous, we will discuss few examples here. Sodium, glucose, co-transport. Now this one is of two subtype, like SGLT1 and SGLT2. Okay, the locations are also different, like this SGLT1, it is located in the enterocytes of digestive system, that is like the intestine, right? In the intestine you have the enterocyte, it is present there. Okay. Now, if there is any mutation to the gene signaling SGLT1, what will happen? What was it doing? in the intestinal enterocyte, it was co-transporting the sodium and the glucose, obviously, right? So if there is any mutation to the gene signaling that one, the formation of this carrier protein will be hampered, it will be distorted, it will not function properly. So it will result in a clinical condition which is congenital glucose and galactose malabsorption. Okay, as a result, the child or the infant, if the severity is more, then it will be very much prominent in the infant stage itself. Infant will have recurrent episodes of diarrhea. Okay, and it can be fatal also if it cannot be corrected, right? This is one. The location of the SGLT2 is located in the proximal convoluted tubules. It is in the proximal convoluted tube. Now, this is the reason why we don't have any sugar in the urine. In a normal person, there is no presence of sugar, right? It is sugar free. That is because this SGLT, this SGLT2 reabsorbs almost 100% of the glucose from the ultrafiltrate. Okay. Now, in this condition, if there is any mutation, if there is any mutation in the gene signaling for SGLT2, then what will happen? It will result in it will result in isolated renal glycosuria. Okay, it will result in a condition which is called isolated renal glycosuria. Now, there are these different drugs which are designed to target all this glucose, sodium glucose transporter. Like the pharma link to it is that, like fluorescein. Okay, it is derived from a plant. This is targeted. It is used to block the activity of SGLT1. Similarly, derivatives of fluorescein, which has like the commercial name as like empagliflozin, ganagliflozin, all those. Okay, so these are used to lock the SGLT2 where to to treat people with diabetes mellitus. Okay, so like SGL2, its main function is to reabsorb all the glucose to the plasma. So people who are suffering from diabetes mellitus, their blood sugar level is elevated. So if this co-transporter subtype like sodium glucose co-transporter subtype 2 is blocked, then it will not be able to reabsorb the glucose, as a result of which glucose will pass out through the urine and the blood glucose level can be maintained. Okay, we'll discuss about few more clinical aspect of the other co-transporters. Okay, the second one is our sodium iodide co-transporter. Where is it located? Now, this sodium iodide co-transporter, this is located in the thyroid follicular cells. Okay, and this is the lumen and this is the blood or the plasma. So here we have this symporter, which is the sodium and iodide symporter. Here actually two molecules of sodium is symported with one molecule of iodide inside the thyroid, inside the thyroid follicle. Okay. Now this iodide will further become iodine, and there is a whole process that will eventually help in the formation of the thyroid hormone. Okay, this is a whole process. Now, if by any reason, if by any reason there is malfunction or deficiency of the sodium iodide co-transporter, then the person will suffer from congenital hypothyroidism. Okay, the person will suffer from hypothyroidism that will be genetically inherited. The pharma link to this is like perchlorate and thiocyanate locks the sodium iodide, sodium iodide transporter. Okay, this two transporter we have discussed. Okay, now comes the third one, that is the sodium potassium chloride symporter. Now, this is a symporter, this is a transporter that you are quite well attached with. You all have idea about where it is. This one is located, it is found where? It is abundant in the ascending loop of Henle, right? It is abundant in the ascending loop of Henle. Now, from the name itself, we know that what it does, it symports like one molecule of sodium, one molecule of potassium, and two molecule of chloride into the tubular cells, right? Now, it is reported and studied that any defect in this symporter, what does it do? It causes Bartter syndrome. And sometimes it is seen this Bartter syndrome is also coupled up with metabolic alkalosis as well as hypokalemia. Okay. Now, talking about an exchanger. Sodium calcium exchanger. So this one, what does it do? This is abundant. It is located in the cardiac muscles mainly. Its function is like highest reported in the cardiac muscle. Though some study says that there is presence of sodium and calcium in the smooth muscle and skeletal muscle also, but it is it is not seen to have any much bigger role or significant role there. It has a very important and significant role in the cardiac muscle. It helps in very easy way, because when we study the cardiovascular system, we will study all this mechanism in detail. So right now, how to remember, it helps in restoring the resting membrane potential in cardiac muscles. Like after action potential will ensure contraction. It will make contraction to happen. So after contraction, for relaxation, for restoring the cardiac muscle back to its resting condition, this sodium calcium exchanger plays an important role. Now, what does it primarily do? Like for this contraction to happen, there is like for contraction, calcium enters calcium enters the cardiac cells or cardiac myofibrils, right? So after contraction, calcium is pumped out from cytoplasm to ECF by this pump. Okay, calcium is removed not only by this exchanger but also by other pump. But here, after the contraction is over, lot of calcium stays in the cell. Some are removed by the Salka pumps and all, which we will study eventually, and some, a large quantity of it is exchanged, is thrown out, pumped out from the cardiac muscle cells, from the cytoplasm into the ECF by this sodium calcium exchanger. Now here the scenario is free sodium is pumped in and one calcium molecule is pumped out by this sodium calcium exchanger. Okay, this also has a pharma link. Digitalis blocks this pump to increase the contractility of the ventricles. Okay, this is how it is important. Talking about the anion exchanger, the anion exchanger, like this is one of the outstanding example in secondary active transport in which sodium is not required. All these examples, and there are many other exchangers and transporter also, like sodium hydrogen, sodium chloride, all of those are there. But in all of them, the common thing is that sodium is a must. Sodium is present, right? But in this anion exchanger, this is like one of its type in which there is no sodium. This is like chloride bicarbonate exchanger. And where it is found? It is located in the RBC cells, RBCs and renal tubules. Okay, in the renal tubules, in the proximal convoluted tubule, distal convoluted tubule, we have this anion exchanger. Okay, it is playing an important role in chloride shift. So chloride shift, for now, let us just remember like it is a physiological phenomenon that happens at the level of the tissue, level of the peripheral cell and lungs, that helps in expelling the carbon dioxide and maintaining the pH. The detailed mechanism, when we study the lungs, the respiratory system, we will study. So for now, you just remember chloride shift or the Hamburger shift. It is a phenomena that maintains the pH of the blood by expelling adequate amount of carbon dioxide. Okay. So this play an important role in the chloride shift. Here, it also within the RBC, it forms band 3 protein in RBCs. Okay. So what is the clinical link to that? Obviously, the clinical link is deficiency of this anion exchanger will hamper the chloride shift, which in turn will affect the pH of the body. Okay. So we have understood the primary and the secondary active transport system. Now we go on to the other type of active transport, secondary active transport, primary active transport, and the third type of transport, okay, which is the vesicular transport. Okay, vesicular transport, just hold on, let me see the no, okay. This vesicular transport, this is by definition here the transportation of biological molecules requires a vesicle. Like, it's not that as soon as the biological molecules are synthesized or like made or manufactured, immediately it is like released. It will be transported via the vesicle. Now, here there are like three types of this vesicular transport. Vesicular transport is of three types. Like phagocytosis, pinocytosis, and receptor mediated endocytosis. Here, this phagocytosis, pinocytosis, and receptor mediated endocytosis, all this three roughly comes under one common thing, which is the endocytosis. Okay, which is the endocytosis. Apart from endocytosis, there is one more type of transportation, which is the exocytosis, where vesicle is also needed, which we will discuss after discussing all these three. To start with, phagocytosis. Now, phagocytosis, which is also called or known as cell eating. Okay. Here, actually, what happens in phagocytosis? It is the debris, or it is like the larger molecule which are engulfed by the cell, So, if we are to draw this one, how does this phenomenon look like? How does this phenomenon happen? So, hypothetically, let us draw this cell. Okay, say this is the cell, and these are the molecules that has to be engulfed in. So first what happens? This is like the cell membrane, and this is the inside of the cell. Okay. So first what will happen is like there will be like an invagination, a pinching or invagination of the cell from its cell membrane side, something like this. Okay, a part of the cell membrane will invaginate or will like have this pinching like configuration from inside. Okay, now this molecules that has to be engulfed, it will sit and attach and bind onto this pinched or like this engulfed portion of the cell membrane. After which there will be total engulfing of this vesicle containing the substance. Like now a vesicle has been formed from the cell membrane which is attached to the cell membrane. A vesicle containing the substance to be brought into the cell is now like sticking out from the cell membrane, and then it will loosen up, get pinched out. It will loosen up and get pinched out and enter the interiors of the cell. Okay, something like this. Something like this. This is the phagocytosis. Now, in this whole process to happen, like binding of the substance for formation of that pit-like substance, engulfing of the substance, and ultimately the pinching of the substance inside the vesicle and then pushing it into the cytoplasm, it needs the support of certain protein. The proteins the proteins mainly involved in endocytosis. Remember, we have spoken about the phagocytosis. The mechanism is same with little difference is in pinocytosis and receptor mediated endocytosis. We will come to that. Now, the proteins mainly involved in the process of endocytosis are the Clathrin, megalin, and Caveolin. These are the main protein which is helping in the process of endocytosis. Now coming to this protein which are of very importance to us, it's like what is actually the protein clathrin. Now structurally this clathrin, it is nothing but it is a triscalon. Okay, this is like this. It has got this domains like that. And there is like heavy chain and light chain attached to this clathrin protein. So here you can see three leg radiating out with a central hub, with a central hub in it. So this is the heavy chain, this is the light chain, and these are the three legs radiating from the central hub. Now, what does this clathrin do? We have seen, right? The molecules is sitting and binding on the cell membrane, and there has been a pinching of the vesicle. So, this clathrin provides support to the vesicle in pinching off or moving off, like this portion, like this one, to move away, to move like from this region to move away towards the inside, for the vesicle to pinch off, move away from the cell membrane to the interior. Okay, in doing so it requires GTP and the supporting protein which is dynamine. Dynamine makes sure that the vesicle that is getting cut off or pinched up from the cell membrane does not breaks apart, does not loses the shape of it. Okay. The clinical link to it is that mutation of latrin is seen in familial hypercholesterolemia. So in this disease, what happens here? The LDL receptor is defective, as a result of which the LDL cholesterol is abnormal, but the triglycerides are in normal range. Okay. Now, what does this do? This causes deposition of cholesterol in the it causes deposition of cholesterol in the tendons, causing tendon xanthomas and premature coronary atherosclerosis. There will be deposition of this bad cholesterol in the coronary artery. Okay. Other important proteins that is important in the process of endocytosis are the megalin. Now, megalin, it is needed for transportation of hemoglobin. These are all taking part in the process of endocytosis. Like what clathrine was doing, this megalin is doing for the transportation of hemoglobin, insulin. Just remember the different name because this all connects to the various clinical condition. Insulin, then vitamin D binding protein, then retinol binding protein, albumin, all this. Okay, so if there is any mutation to megalin, means to the gene that will ultimately result in a defective megalin, it will result in a syndrome which is called as Donnai Barrow syndrome. So, what will be the characteristics here? And of course, this is a congenital because there is a mutation. Whenever there is any mutation and signaling of a difficult or a deficit or a inefficient protein, this is always congenital because anything that happens at the level of the gene is always hereditary. Okay. So, here, here, the child will have defective corpus callosum. Now, corpus callosum, it is the structure that is joining, that is a connection between the right hemisphere and the left hemisphere of the brain. So, if the connection is not proper, then the coordination and all the neurological function and all the development of the brain and associated structures will be defective. Corpus callosum. Then there will be hernia of the diaphragm. There will be ocular anomalies. because the structure in the brain are not developing properly. So everything that is related to the face, facial structure, vision, facial formation of the facial characteristics, hearing, vision, everything will be affected. Ocular anomalies, there will be like facial disformation, there will be hearing loss. All these characteristics will be manifested. Another protein is the Caveolin. Now, these are all the very recent protein that has been reported that participates in the process of endocytosis. Now, Caveolin, it is related to transportation of a very essential cofactor, which is cobalamin. Okay, which is very important, essential factor, cobalamin. Okay, or cobalamine. So it is helping in the transportation of this cofactor. Now, if there is any mutation in tubulin, results in a condition called Imerslund Presbeck syndrome, and this will aggregate, which will it will then aggregate into megaloblastic anemia. Okay. So, three proteins in context to the endocytosis we have studied. Apart from that, very important protein which helps in the same is our caveolin. So, let me first tell you what is caveoles, because the term caveolin connects to caveole. Now, we have studied that cell membrane, it is a our cell membrane, it is a layer of lipid protein bilayer, right? Following the fluid mosaic structure. Now, the cell membrane, there are some areas. There are some areas in the cell membrane which is like rich in sphingolipids. Okay, which are rich in sphingolipids. Now, such areas, such portions are called as lipid rafts. Like it has got only lipid and sphingolipids, has got lipids and sphingolipids. Now, such lipid rafts, okay, this such lipid rafts may be linear or may be pitch shaped. Okay, maybe linear or maybe pitch shaped. So, some cell membrane, like some region of the cell membrane may be like linear, which is rich in the lipid and sphingolipid, and then all of a sudden it may undergo invagination like this, like pinching and invagination like this. Now, this structure that has pinched in is called as a claviole, and the protein which is helping in this caveole formation and transportation is the caveolin. Substances like folate, okay, like folate, folic acid, folate, uses caveolin to transport. And folate is very essential for the formation and maturation of the RBC, because without folic acid, there will not be incorporation of the iron. The RBC will not be matured. It will not have this biconcave shape with the correct diameter, and then it will result in anemic condition. So folate uses this caveolin for transportation. Apart from that, certain toxins like cholera toxin, papillomavirus toxin, they use this caveolin protein to get endocytosis, to get endocytosized, means to enter the cell. Okay. The pharma link to it is that antifungal drug nystatin blocks the activity of caveolin proteins. Okay, these are the important characteristics of the protein involved in endocytosis. Now that we have understood what is endocytosis, pinocytosis is also very easy. So very quickly we can write what is pinocytosis. Like pinocytosis is like cell shrinking. Like the diagram is saying, the process is saying, instead of like what we have seen here, the same thing happens. Like this is the same thing. Instead of the bigger molecule, the molecules that will be entering will be smaller in size. Okay, that will be smaller in size. So by this small molecules like amino acids enter the cell. Okay, that is our pinocytosis. Next one we mentioned was the receptor mediated endocytosis. Here also it is the entrance of a molecule, bigger molecules. But the only difference is that the molecules that will enter the cell, instead of directly sitting on the cell membrane, they will first sit on the receptor. Certain molecules, especially hormones, they're bigger in size, right? So what happens? So let us again draw the hypothetical diagram of the cell. So what happens in this cell? So this is the cell. So here there are certain receptor that is facing the ECF. Certain receptor is facing the ECF. This is your cytoplasm. Okay. So when this molecules comes, they first bind onto the receptor. Once they bind onto the receptor, what will happen? Again, now here the invagination will be of the molecules that is bound onto the receptor. So, so together with the molecules bound onto the receptor, there will be invagination, like moving of this structure inside the cell. Like this. The receptor along with the molecule will now face the cytoplasm ECF along with the receptor bound on it. Then after that, as usual, there will be detachment. There will be detachment of these molecules from the vesicle. This thing will lyse out. Ultimately, this molecule, it will like lyse out, rupture out of the vesicle and then come to the cell. Okay, this is all about our endocytosis. Another process which is a type of vesicular transport is exocytosis. So by the name itself, we can understand exocytosis. It is just opposite of endocytosis. In endo, the molecules entered the cell. In exocytosis, the molecule will actually leave out the cell. Okay. So this molecule which is leaving the cell or the process of exocytosis, it is also called as cell vomiting. Okay, it is also called as the cell vomiting. Like substances will be lysed out or thrown out of the cell. So here you see the most important thing to remember in case of exocytosis is that through exocytosis or by this process, insulin, the very important biological molecule, hormone, insulin, and neurotransmitters are secreted. Okay. Another interesting, another interesting and essential point to remember, I'm putting a star in it. We can't forget it. It's very, very important, which is that this this is a calcium dependent. It is a calcium dependent process. Exocytosis cannot happen without calcium. Without calcium. The reason is this, which is, see, just the general idea, then we will come to the details of the exocytosis process. Why calcium is essential for the process of exocytosis. So see this. Here, the cell membrane. Okay, say this is the hypothetical diagram of the cell and this is the cell membrane. So what do we know? This is our cell membrane. Okay, I'm just making it thickened instead of drawing two lines. This is our cell membrane. What do we know? Cell membrane, it is, what is it? Cell membrane is negatively charged. Cell membranes are negatively charged, right? Now, exocytosis, it also requires a vesicle. Like this is the vesicle which will carry the substances that has to be thrown out of the cell. Okay, so this will ultimately go there and it will use here, just the opposite of the endocytosis. It will use here, right, with the substances, and then it will be lysed out, right? It will be lysed out to the exterior, right? Now, what role does calcium play here? What role does calcium play here? See, this cell membrane is negatively charged. Okay. Well, what is this? This is a vesicle. This is a vesicle, right? Now, this vesicle means what? It is a sac that has a membrane again. So anything that has membrane will have the same composition like that of the cell membrane. So this is also negatively charged, right? So when two negatively charges are approaching each other, like when this is for our understanding, quite little down. Like say this is the cell membrane negatively charged and this is the vesicle membrane, okay, negatively charged. So when both are negatively charged, both negatively charged will repel each other, right? So what calcium does is calcium forms a bridge. So here calcium, which is positively charged, right? Which is positively charged, makes a bridge. Okay, it kinds of makes a bridge and it kinds of attaches this vesicular cell membrane to the cellular cell membrane, helping them to attach and then lyse the substances. This is why calcium is essential. Now, what is the process, like what is the importance of the process exocytosis? Exocytosis is used for both to our advantage as well as to our disadvantage also. Now, the advantages of exocytosis, obviously, what is that? Those are the neurotransmitter secretion, especially in the neuromuscular junction. Everywhere we need like neurotransmitter like acetyl-CoA. It is so much important for generating action potential, for contraction of muscles of our limbs, for carrying out the motor activity, right? Neurotransmitter secretion as well as insulin, very important to maintain the blood sugar level, right? Secretion. Okay, this is exocytosis. It's not only that in this two function exocytosis is there, but these are the two main functions or very prominent function of the body which requires exocytosis. Okay, these are the advantages. Now, what are the disadvantages? It also holds some disadvantages as we discuss. The disadvantages of this exocytosis are that toxin like Clostridium botulinum and Clostridium tetani targets snare protein. Okay, I'll leave this gap for talking about snare protein. Snare protein, which is essential for exocytosis. Okay. Now, what is this snare protein? Snare protein, the full form is soluble in ethyl melamide, A factor sensitive protein receptor. Okay. Now, this is essential for exocytosis. Now, what does this Clostridium, the toxic or toxin from this Clostridium botulinum do? This botulin toxin destroys snare protein and induces flaccid paralysis. Little in depth to this. So, see, this is the snare protein. Okay, it has got it has got the subtype which goes by the name synaptotagmin. Then there is syntaxine. Then we got snap 25. These are all the subtypes of the snare protein Okay, and then we got synaptotagmin, all this, and many more. These are the few important ones to mention. Now, this botulinum toxin, it got like A, B, C, D, E, F, and G subunits. Okay. Now, through this A, B, C, D, and E, F, and G subunit, it it can systematically target like different subunits of the snare protein. For example, for instance, like syntaxin is blocked by the C subunit of the botulin toxin. Then synaptorevin is blocked by E, T, F and G subunit. Similarly, the SNAP25 is blocked by the A and E subunit. Okay, like that. So it is also banned from using in the warfare, because earlier when it was used in warfare, it would just block the activity of snare protein. So all this subunit, once the toxin blocks the snare protein, then acetyl CoA cannot be released. So if acetyl CoA in the neuromuscular junction, that also we will study, it is something like this, right? This is the axon of the neuron, and this is like the sarcolemma. This is like the roughly the neuromuscular junction. So here actually the acetyl CoA is released, which then binds onto the receptor on the postsynaptic membrane, and then action potential is generated. Now, if this one, the whole process, if this one is like blocked, if it is blocked by toxin, because this, if the snare protein which is required for the exocytosis, if this is blocked, then there won't be release of the acetyl CoA, and without acetyl CoA there will be no action potential generated. Not only there will be no action potential generated, in the neuromuscular junction there is also certain acetyl CoA that always stays, which makes our skeletal muscle tight. So if snare protein is blocked, the whole process of release of the acetyl CoA will be blocked, cannot happen, as a result of which there will be loss of the tightness also in the skeletal muscle, which will induce flaccid paralysis. Now, sometimes this is used in bioterrorism also, but internationally in the warfare, this usage is blocked. Usage of this botulin toxin is blocked. Okay, this is the one toxin that is blocking the snare protein. Another toxin that blocks the snare protein is the Clostridium tetani. What it does? It induces spastic paralysis. Now, this is again very different from how the botulin toxin was working. Okay, so what will it do? It will induce spastic paralysis. How does it do? Here, just before going into spastic paralysis, one thing, the spinal motor neuron, our spinal motor neuron innervates the motor muscles. That is basically the muscles of the limbs and all. Okay, that is involved in the motor function. So once there is contraction, once there is firing from this motor neuron, immediately the Renshaw cells inhibits the motor neuron. Like once the firing is done, contraction is happened, the Renshaw cell will inhibit the motor neuron so that relaxation can happen. Otherwise, there will be constant firing from the motor neuron and there will be like constant contraction, which is not at all desired. So, Renshaw cell will inhibit the motor neuron after firing from the neuron. Okay. On other side, GABA and glycine also on the other side, GABA and glycine also use snare protein. Now, GABA and glycine, they are the inhibitory neurotransmitter. Acetylcholine was the excitatory neurotransmitter. GABA and glycine are the inhibitory neurotransmitter. They will always stop the formation of action potential. Okay. So, this Clostridium tetani blocks the release of GABA and glycine by blocking snare protein. So, if snare protein is blocked, then right after the firing from the spinal motor neuron, the release of the GABA and glycine will be stopped. There can't be release of this inhibitory neurotransmitter, GABA and glycine. As a result of which, what will happen? There will be more and more firing. There will be recurring firing from the spinal motor neuron. Now, if there is constant firing from the spinal motor neuron, what will happen? It will result in spastic paralysis. Means there will be spasms of the skeletal muscles and there will be paralysis. Okay, this is how it works. So, in today's lecture, what did we learn? We learned in like total details about the secondary active transport with examples, its clinical link and pharma link, and also we understood the vesicular transportation in detail. Tomorrow we will start with the cell physiology. We will start with the cell membrane, characteristics of the cell membrane, and essentially the clinical links to that. Like what would happen if some important constituents of the cell membranes are missing. As I told you, in each and every lecture, we will go into minute details so that you have a very good understanding and concept for your exposure to the clinics, especially pathology and intro. Okay. So Today we are going to wind up the session here, and I will see you all tomorrow then. Okay, thank you.